Alright guys, welcome back to Underground Science, and in this video, we'll be talking about graded potentials of a neuron. Alright, so let's go and get started. So we're kind of going to try to build up, maybe in the next video or the video after that, we're going to talk about action potentials and the step of an action, like each step of an action potential and what's actually happening in the neuron. And we'll see the graph for that also. Alright, but in this video, let's go in and see what graded potentials are for a neuron. Alright, so let's go and start out by drawing a structure, or the basic structure of a neuron. Alright, so let's go and draw our cell body, or our soma, right here. And actually, let's draw a little bit over here. There you go, right there. And so this is going to be our cell body, and then we're going to have a nucleus, right? In the nucleus, we're going to have our DNA. So our DNA in the nucleus. And of course, we're going to have other organelles, but just to save time, we won't list all the organelles out, but maybe I can just go and write down like a mitochondria here. Alright, just like that. Hopefully that looks like a mitochondria. I mean, it probably doesn't because I drew it too small. But anyway, so that's our soma, right, of our neuron. And then let's go and draw our projections, or our dendrites. Okay, and our dendrites are super important, right? Because without our dendrites, we wouldn't be really able to receive information or electric signals from another neuron that well. So let's go ahead and draw our dendrites in, our tree-like branches. All right, so let's go and draw a few more in. And all right, I guess that's good enough. Let's go and draw a couple more in. All right, so these are our dendrites, and then remember that we have our axon hillock, or our trigger zone, right in the beginning of our axon. So it kind of like branches out like this, and we have our axon hillock right about there. And then we have our so let me just go and, like, I don't know, guess shade in like this, just representing the axon hillock. Trying to guesstimate the exact area is going to be about right after the soma, or in the beginning of right before the axon really starts. And then we're going to have the axon of the neuron, right? We're going to have the axon. This is where the action, oops, dang it. This is where the action potentials will travel down, or electric. Just think of action potentials as electric signals traveling down an axon. Okay, so if someone asks you what does action potentials mean, right? Whenever you're describing a communication through an axon, all action potentials are just electric signals traveling down an axon so that you can communicate with another target cell or another neuron. Right, so this is our axon. Okay, and then at the end of our axon, we're gonna find a structure called our axon terminal, and because our axon is terminated now, we're done with our axon. Right, and what's gonna happen is we're gonna actually end with another structure here. This is gonna be our synaptic bulb, or a synapse as a whole. Okay, so we're gonna have our axon terminal at the end of the axon, and then this whole structure right here is our synaptic bulb, right? Or you could just call the whole structure as a whole as your synapse. You're synapsing on another target cell or another neuron, okay? So actually, you know what? Let me just go and label it out. But make sure to go and check out my basic structures of a neuron video, because that's where I discuss all these, except for our axon terminal and myelin. We're gonna talk more about that when we talk about action potentials. Right? So these are our dendrites, right? This is our cell body, or soma, of our neuron. Try to label this as our axon hillock. And remember that this is also called our trigger zone, because this is where our action potentials actually start, or where it's generated, okay? In red, we have our axon, where it's going to actually travel down. The action potentials are going to travel down the axon. And just know, in some nerve cells or neurons, right, either in the central nervous system or the peripheral nervous system, some nerve cells will have myelin around it. And myelin is pretty much just mostly made of lipids and a little bit of protein, right? And myelin is there so that we don't have to conduct our action potentials down the axon slowly. Myelin helps us conduct our action potentials and make the signals and electric signals travel down the axon way faster and much more accurately. Okay, so it makes it much more efficient for the signals to travel down the axon. All right, so these are our myelin sheaths. Okay, and these are myelin, and we'll go over glial cells in another video. Glial cells of the nervous system, central nervous system, and peripheral nervous system, and one of those glial cells in each central nervous system and peripheral nervous system is responsible for the production or synthesis of myelin. All right, so we'll go look into that in a future video. But just know myelin is there on some axons, and if we do have myelin on an axon, like usually people refer to as myelinated axons as white matter, and unmyelinated axons or unmyelinated axons, right? If there's no myelin, we refer to that as gray matter. All right, so if there's no myelin on a group of axons, it's pretty much gray matter. And if there is myelinated, if the axons are myelinated, then we refer to those neurons that are myelinated as a whole as white matter. Okay, but that's just good to know, I guess. So myelinated axons are white matter, non-myelinated axons are gray matter. Okay, and so let's go ahead and see what graded potentials are, and for that, we'll have to zoom in into the dendrites, all right, because that's where it mostly happens. Okay, so let's go and zoom in into the dendrites. So let's go and draw like a little zoom in sign here, try to at least. And we're going to go all the way down here and just use this whole space as referring to the dendrites. So let's say, let's just draw the dendrites as a huge thing right here. And and we're zooming into the soma and cell body and the axon hillock. Okay, so let's go and do that. All right, so we're going to have our huge dendrites here. So what I'm just going to draw like 2 or 3 dendrites, all right? So let's go and draw, actually, you know what? I'll just draw 1 dendrite here. Just a huge dendrite. Okay, just a projecting out dendrite. Okay, that was really bad. Sorry about that. Let me go back. All right, just a huge dendrite here. And we're going to draw our soma, or our cell body, right, like so. And then we're going to draw our axon hillock, like so. Right about here, just before, I'm just trying to like show it that it's before the axon starts, because the axon hillock is called the trigger zone because that's where the action potentials are generated. Okay, so that's where the action potential is generated, and then those action potentials will travel down the axon afterwards. And we'll see that in this, I think in this video, if we go and talk about it, that the axon potential is only generated at a certain point. It's an all or none phenomenon. So if it reaches a certain millivoltage in the axon hillock, then an action potential would be generated. If it doesn't, then it won't be. All right, so let's say that there's another neuron, right, synapsing this whole postsynaptic neuron. So let's say there's another presynaptic neuron over here that has its synaptic bulb and its synaptic cleft, and it's sending neurotransmitters over. All right, so just the basic idea is that when that happens, right, when the neurotransmitters are bind to these dendrites, this whole dendrite right here, this is draw 1 right now, is that let's say we have some amount of positive charge that's generated, okay, from the neurotransmitters binding to these dendrites in this neuron. Okay, so let's say we have some positive charge generated here on the dendrites. But these positive charges are called, whenever you have a positive charge, you'll see on a graph that you're getting more positive in terms of millivolts. Okay, and if you get more positive, that's called depolarization. And on the other hand, if let's say we're not getting, so this will represent a stimulatory signal, right? So this is a stimulatory and excitatory, or an excitatory signal. So excitatory. But we don't always have to get an excitatory signal, right? What if we don't want to fire this neuron right here? Then what's going to happen? We're not going to get an excitatory signal. We're going to get an inhibitory signal. If we get an inhibitory signal, we're going to drop in terms of total charge inside our neuron. So we're going to have not depolarization, but we're going to see graded potentials of hyperpolarization. And hyperpolarization is when the neuron goes below its resting membrane potential. All right, so pretty much we'll discuss this in the next video about how an action potential graph looks like. But pretty much, right, this may not make a lot of sense right now because we haven't talked about an action potential graph yet. But pretty much inside our neuron, we have millivolts here, okay? This is our membrane potential in millivolts. And then we have time here, all right, in milliseconds. And so what happens is that if our neuron gets a lot of these excitatory signals, then we're going to see that okay, first let me draw this. So we have our resting membrane potential here. Our resting membrane potential is about negative 70 millivolts, all right, of our neuron, negative 70. So if our neuron's not firing, it's here, right? It's in its resting membrane potential. But the second we get so many excitatory signals that from negative 70, if it hits about like negative 50 millivolts, if it hits that, right? So let's say it's getting more positive, it's getting more positive, it's getting more positive, right? Because of all the excitatory signals. And see, we are getting positive as we go up because remember this is negative. So as we go more up, we're getting more positive. So we're getting excitatory signals, and oh, we hit the threshold potential, right? So the threshold potential is in the range of about negative 45 to negative 50, got arrow here, to negative 50, no, it's confusing. Go and erase that real quick. All right, so our threshold potential is about from negative 45 to negative 50 millivolts. I showed negative 50 millivolts in this graph, okay? As you guys can see right here. So again, all these excitatory signals, and we'll talk about that it's not just like the signals don't just come on the dendrites and then you suddenly start getting like excitatory signals on the graph, okay? But we're just trying to get the basic idea here. So we're getting excitatory signals, and bam, we hit the threshold potential of negative 50 millivolts about, okay? And right then and there, that's where the all or none phenomenon comes in for an action potential. No matter how strong the signals are, or even though your action potential somehow reaches negative 50, it's going to spike up. All right, it's going to spike up in positivity in terms of our millivoltage, and it's going to go to about negative 35 millivolts or positive 35 millivolts here. All right, so it's going to go to about positive 35 millivolts before it drops down. And it's going to drop down. We're going to see something weird. It's not going to go back to its resting membrane potential. It's going to go even more down until it reaches about negative 85 millivolts. And then it's going to slowly rise back up, and it's going to end back at its resting potential, ready to fire again. So this is how an action potential is. It's just the basic idea of it. We'll look more into it in a future video. All right. So again, this negative 70 millivolts is our resting membrane potential. So meaning that's what our membrane potential is when our neuron is not firing. Okay. So resting membrane potential and our negative 45 to about negative 50 millivolts, is our threshold potential. So that's the limit. Once it reaches there, no matter what, it's going to fire all the way up to about positive 35 millivolts. All right, so this is our threshold potential. All right, and then once it comes down, it's actually going to come down to about negative 85 millivolts. All right, and we'll talk about our refractory periods and hyperpolarization, all that in the next video. But what I want to say is that when many of these positive signals that are coming are going to be called graded potentials. All right, these are going to be called graded potentials. And what this means is that just because one positive signal, let's say we have a very excitatory signal that comes in, all right, and we have some depolarization going here. Okay, so that's how you represent depolarization because we're getting more positive here. In terms of this graph, like right here, if you drew it, this would be our depolarization state, then we would have our repolarization state, then our hyperpolarization, and back to resting membrane potential. All right, when we'll talk about those stages later, I'm getting off topic here. So we're having excitatory signals here, but what happens is that these excitatory signals actually travel down the dendrites and through the cell body of the soma, and they actually reach the axon hillock. But by the time they reach the axon hillock, if this positive signal was like huge, right? This guy was huge, like this big. Okay, let's say the positive signal was this big, the excitatory signal. By the time it gets down to the axon hillock, our 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 size and our magnitude of that signal, of that graded potential, is going to decrease a lot. Okay, it doesn't stay the same. So that's why just because you get a huge positive excitatory signal on the dendrite doesn't mean we're going to reach the threshold potential here. Okay, because our size of our graded potentials, I couldn't speak there, decreases as we get down to the axon hillock. All right, so by the time it gets here, the graded potentials are weak, all right? They're not that strong. So that's why you need a lot of excitatory signals on our dendrites right here in order for our graded potentials to be positive enough here so that we can hit our threshold potential, which is right here in our graph, and so we can have an action potential fire down the axon. All right, so we need enough of a membrane potential at our axon hillock, not on our dendrites, because again, as we go down, the magnitude and the strength of that signal decreases. It went from huge, like I'm about to like do this, right, to oh crap, I can't fire a signal. So we need a lot of those excitatory signals. Same thing works for inhibitory signals, all right? By the time our inhibitory signals are huge here, by the time it gets to the axon hillock, it's not really that huge, all right? It's not that strength. It's not that strong, I should say, in terms of making that neuron not to fire. Okay, so if we're talking about excitatory signals or graded potentials, we need a lot of excitatory signals on our dendrites in order to reach our axon hillock, and for our axon hillock's membrane potential to be about negative 45 to 50 millivolts, right? So we can hit that threshold potential right here and then get our axon to have action potentials travel through it. All right, so those are pretty much graded potentials, okay? And we'll talk more about graded potentials in the next video, and also about action potentials and the step-by-step process, all right? So we have a lot to learn, and so hopefully you guys tune in to my future videos about biology and physiology and biochem, and physics. Really hope to see you guys there. Don't forget to hit that notification bell and subscribe and like, and see you guys later. All right, and stay safe.